I am very pleased to introduce Miss Cheryl Sesler. I was lucky enough to recruit her at one of the ASB meetings. It's been a couple of years ago now that we first met each other at the ASB meeting. And at the time she was doing gut microflora work on woodpeckers, with a particular type of woodpecker. And that's what she wanted to continue doing when she came here, but unfortunately the person who started the research at UT Martin, where she came from, didn't want to let the research go. So we had to pick another topic, and she was interested in amphibians, frogs, and birds. And so we decided to do something with frogs. She's going to present her research on the adult frogs, and then maybe later on we can get another student interested in doing the tadpoles, and I'll let her tell you more about that. But she's been a fantastic student, hard worker, and she's worked very independently. So I'm pleased to let her present her thesis research to you. Thank you. Oh, afterwards, after her oral test after this, I've got a little cake for her. And one other announcement, we have a tick symposium from 1 to 3 o'clock this afternoon over in the nursing building. So please, everybody that can, come and attend. We've got some big names going to be presenting their work.
Okay, like Dr. Blair said, my name is Cheryl Sesler, and I'm going to present my thesis work on the molecular identification of gastrointestinal bacteria from a sample of adult Cope's gray tree frogs. So just to kind of start out a little bit with a little background about amphibians, because frogs are obviously amphibians, hopefully you know that. It was kind of first brought to the attention of a bunch of scientists at the first meeting of the World Congress of Herpetology in 1989 in England that amphibians were declining in populations. And they've noticed they had noticed the trends over many, many decades. And they actually noticed that the rates of decline were occurring faster than would normally be predicted based on predator-prey interactions and inter- and intra-species competition. And they also noticed that these declines were occurring within all different types of habitats, and that includes aquatic, semi-aquatic, terrestrial, arboreal, and fossorial. And one study was published on the freshwater habitat indices, where they looked at about 200 different types of organisms that live in freshwater, as these frogs will live in freshwater. And those 200 species were not all amphibians, but they did include amphibians. And they looked at the populations from 1970 up to 1999, and they found that there was actually a 50% decrease in the species of organisms that they looked at in these freshwater tropical. So some of the factors is actually more extreme even than tropical rainforests, which I thought was a very interesting fact when I read about it. So some of the factors that are involved in this population decline of amphibians include habitat destruction, pollution, diseases, climate change, and ultraviolet radiation. Now, most of the time these factors work together. It's not just one factor causing it. Because even in protected habitats where habitat destruction is not an issue, population declines are still occurring. So it's something more than just one or two factors. So the organisms that I chose to study in my thesis were Hylocrisoceles, also known as the Cope's gray tree frog. Now these frogs are primarily found in the eastern United States, and they inhabit wooded areas, usually near water, so ponds, pools, and even slow-moving streams. And the adults are insects. They eat insects. They forage on insects. Tadpoles primarily forage on algae, plankton, any kinds of plants in the water. So there are a couple of reasons why I chose Hylocrisoceles as my study subject. One was because it was abundant at the time of the study in this area. But the main reason why I chose it, above the other ones that were here at the same time, is that Hylocrisoceles has actually been shown to be tolerant to many of those factors associated with population declines. So there have been very few reports on diseases, specifically chytridiomycosis. There have been no reports as far as I've been able to find. And then they're also resistant to different pollutants, and habitat destruction has not seemed to be an issue. Now, there has been one study that I found that has indicated that, or has shown that Halocrosophilis is on the endangered species list in New Jersey. But my opinion is that the reason why it's on the endangered species list is because it's very closely associated with another species, Haloversicolor. And for a long time, these two were considered the same species. And it's been until recently that they've actually determined that these are two different species. The only way you can tell them apart is based on their call types. Halocrosophilis has a slower trill than the Haloversicolor, and also based on their karyotypes. Halocrosophilis is a tetraploid, while Haloversicolor is diploid. So when you look at the two organisms, you see that Halocrosophilis is primarily found in the southeastern United States. Haloversicolor is in the northeastern United States. So it would make sense that these populations are considered in decline in New Jersey because it may not be part of their normal distribution. Large transition throughout their life, where they transition from the larval stage to an adult phase. And this transition is known as metamorphosis. It's stimulated by the thyroid hormone. And it's during this transition that the anatomy and physiology of the frogs are altered, both externally and internally. And some of these alterations are very extreme. And this does include reabsorption of many different structures, like the gills and like the tail. It also includes the remodeling of different structures and organs, such as the brain, the liver, and the intestines. I'll talk specifically about the intestines in a few minutes. And then also the development of new structures, like legs. So looking specifically at the intestinal physiology, there are two primary functions for the intestines. This is for any organism. They're there for nutrient assimilation, so you take in food, you break it down, you get nutrients from it. And then also they're there for immune function. It is very closely associated with the gut, associated with lymphatic tissue, which is actually the site of a large number of immune cells in the body. Actually, the largest number of immune cells in the body are found in this gut associated with immune tissue. But one thing that's very important to know is that the guts, the intestines, do contain a large number of symbiotic microbes. And you can see in this diagram, this gray shaded region in each of these, which I will describe in the moment. This is where those symbiotic microbes do exist. And a lot of scientists, because these microbes, there are so many of them, and because they do have they have critical functions within the intestines, they're actually considered an organ within the intestines. I mean, basic cross-section of the intestinal tract. I want specifically the adults first because that is what I study. And this is a general diagram of what you would see in cross-section. You have these circular folds within the intestinal tract, which is going to allow for more surface area for the food to come in contact with the lining of the intestines, as well as for attachment sites for those microorganisms. The food and microorganisms will be within this gray shaded area, like I said before. Now I want to compare this to the tadpole just for a moment, just to kind of show you how much they change during that transition from larval to adult phases during metamorphosis. This is what the tadpole cross-section of the intestinal tract would appear. They only have one single fold. Very interesting in my opinion. The tadpole intestinal tract is much longer than the adult intestinal tract, and that's primarily because it needs a longer area or more area for fermentation, because they do eat more plant-based materials, so they do need to degrade something that's a little harder to degrade. I'll kind of talk about in a minute too. So these microbial symbionts, they are very essential to the proper function of these intestines, like I mentioned. But it's very important to note that these communities are constantly changing. You do have organisms that have specific niches within your intestinal tract, but then you also have some organisms that are simply on the food that you take in, or in the liquid that you take in. And they just simply pass through the intestinal tract. So they are present, but they're just passing through. The organisms that do provide that niche within or do have a niche within the intestinal tract, they're there, they help provide additional oxygen for the host organism. They produce, or they provide usable energy, amino acids, and vitamins, such as vitamin B12 and vitamin K. And again, this is for the host organism as well as for the other microbes that are within the intestinal tract. And then one of the primary functions of these microbial symbionts is to aid in the breakdown of complex carbohydrates, such as chitin and cellulose. chitin is the one of particular interest for me because our frogs do eat insects. Insects have the exoskeleton that's primarily composed of chitin. So we would expect to find organisms that have the ability to